Okay, good morning. So what we have finished in last lectures is like we talked about DNA, right? And then how it, and then we learned about many things about it, and how it was like the genetic material of it. But what we will look today is the what is called as central dogma of molecular. Right? I'm sure you have heard of it. Central dogma tells about how DNA actually goes into action. Right? What DNA can do. Right? So DNA can actually is responsible. Right? We know that DNA contains the genetic information. We have lots of DNA in one cell. Almost if you put DNA end to end, it's like 6 feet long. Right? Imagine it's only a molecule, right? So 6 feet long, and then 10 trillion cells are there in your body, right? So if you put all that DNA together, right, I'm sure it will go many times from Earth to Sun and back, right, several times, right? So it is that long DNA that you have in your body, right? And of course, like there are like in one genome, we know that there are like 3 billion bases, 3.17 something, but on an average, if we consider, it's like 3 billion bases. So 3 billion bases of DNA, and that is where what is an information written into, right? So if you put all the sequence of DNA into a book, right, it will run into volumes, right? That just your sequence, right? Among that sequence, there are genes, there is genetic information. Last time we talked about traits, isn't it, right? The human face traits, right? You are looked into that, right? And then every trait is governed by DNA, right? We know that. So there is a genotype and there is a phenotype. So if we say I have a different hair color, if that's a phenotype, right? But then there got to be a genotype behind it, right? So genotype-phenotype relationship is there, and that is what we definitely know about, right? That is what we have learned, right? But then how does genetic information flow? One information flow, what we always talk about is vertical. Vertical means what? From parents to their offsprings, offspring to their offsprings, right? From generations and generations after the information flow. This is what is called as a vertical flow of genetic information. There is something called as a horizontal flow of genetic information, right? What is a horizontal? Which is like from like bacterial conjugation or transduction or transformation. Right? If like we look into that, that's a horizontal flow of information. Meaning what happens is in that case, it is not through the sexual cycles. Right? Out of that, if we could get an exchange of genetic information, that's a horizontal flow. But we are not talking about that flow of genetic information. What we will be talking about, the flow of genetic information from DNA unto RNA, and from RNA into protein, right? And from protein to even goes into metabolites, right? And metabolites to goes into traits. That's what we have seen last time as well, right? So what is the central dogma? Central dogma states simply that it is the information that from DNA, right? It goes to RNA, right? From RNA it goes to protein, right? And from protein to metabolites, and from metabolites it goes to traits, right? But classically, the central dogma relates only from DNA to RNA, RNA to protein. And in many books you will find that in DNA you have a circle like this, right? Which represent DNA replication, right? So it was considered that a DNA replicates itself, but that's not true, right? DNA cannot replicate on its own. What DNA would require is DNA would transcribe protein into RNA, RNA to protein, and proteins are those which will be responsible for DNA replication. Right? DNA cannot replicate just spontaneously, right? Unless and until it has those many proteins. We will look into DNA replication in details. That time you will see that there are many proteins that are actually involved into DNA replication. right? But then unless and until there are proteins, it will not go into. So the workhorse in our cell systems are proteins. Right? We have enzymes, we have receptors, we have got the molecules, right? We have got so many different things that would work together in cell transduction. If you look into that, a cell would transduce signals with the help of so many different proteins. Like kinases and kinases and kinases are there, which would like, so there are like kinase, and so you have like look into that. We will look into that down the line. But then there is a good flow of information that happens within the cell, and then proteins play into a big role in it. So proteins work, proteins are the structurally important, they are functionally important. There are certain structurally important proteins also, which like cytoskeleton proteins, which gives you which gives the cell its own shape, size and function, right? Of course, right? So those are those proteins which are structural proteins. Histones, for example, are structural proteins which bind to DNA and mix it, condense it into a form of chromosome, right? So histones proteins are structural proteins, whereas there are certain proteins which are enzymes. The exonuclease that we talked about, right, are functional proteins. So all enzymes are functional proteins. So proteins function in a way, right? So everything happens at the cell level is through proteins. So proteins are so important, right? But then proteins are made up of what? Proteins are made up of amino acids, right? Individual amino acids put together engage into polypeptide or peptide bonds, right? And then that's why we call the protein as a peptide chain, the chain of peptides, right? And this chain of peptide then folds, right, acquires a three-dimensional structure, and that's why it will be serving as a particular enzyme, right? So it has some kind of function. So there is a structure-function relationship we talked about in earlier lectures, right? So but then this protein sequence, so who would determine protein sequence? It comes from the sequence of RNA. And where the RNA sequence comes from? That comes from the sequence in the DNA. So sequence in DNA, right, give rise to RNA. When we take RNA, it is what we basically are speaking about our messenger RNA. And this messenger RNA with the help of rRNA. rRNA is what? Ribosomal RNA and tRNA. That is transfer RNA. So these two RNAs, this messenger RNA, so all these three RNAs together makes the protein synthesis machinery in eukaryotes, right? Finally they make a protein, right? So now what this process is called from DNA to RNA? Right? Transcription, right? What is called as transcription? And this is called as translation. Right. Now pay attention to these words, transcription and translation. Transcript means what? The change of script. Right? The change of script is transcription. Now what is the change of script here? The script is the DNA sequence. Right? The script is DNA that is changed to RNA. But the code remains the same. Right? So it is only transcripted. The only change is what? The script is changed. It is like when you type a SMS or message, right? I am sure you do every day, right? You write it in English, but maybe the sentence is in English, in Hindi potentially, right? You ask your, I am waiting for you in canteen, but you don't write in English word, right? मैं canteen में हूँ, right? If you write it likewise in English, so the script change, but the language is Hindi, isn't it? Right? Or in Marathi or in any other language for that matter. So what you are doing basically, you are writing Hindi in English script, right? So it's a kind of what? You are changing only script, right? So the script change from DNA to RNA, that's why it is transcription. This is something easy to remember, that's why this is what it is. But if you want to translate something right? If you want to translate, I am in the canteen. If you want to translate into the Hindi, then what will happen? It will not only change the script, right? You will have to change the grammar too. So the grammar would also change because Hindi has a different grammar, right, than English. Now it is translation, right? So from RNA to protein, it is translation. It gets translated into why? In a sense, why? How it happens? See, DNA and RNA are chemically very similar, right? But protein are different, right? So in DNA and RNA both have the only difference. There are some small difference between DNA and RNA, right? D-ribose sugar, deoxyribose sugar, and all those you know that. But then basically the same thing, right? You have the backbone, nitrogen bases, right? The same thing there. But then when it comes to protein, the protein is a complete different chemistry, right? Chemically, amino acids are completely different entity. They have a different bond, right? That's a polypeptide bond, which is not there in any of this RNA or DNA. So then its grammar is different, right? That's why it is called as translation. So it is translated into the protein, right? So this is what two words you have to remember: transcription and translation. Now there are of course like lots of details in transcription. We will go into that when we will study that in details. But then in transcription, what happens, right? All this mRNA, especially in eukaryote, we are talking about, right? I have seen, like we have seen the difference between prokaryote and eukaryote. Prokaryotic genes are continuous, right? But eukaryotic genes are interspersed. Interspersed means what? Between coding and non-coding regions, right? So now what will happen if this messenger RNA is made from the DNA? What will happen to that? It will has to undergo changes, right? What you have to do is you have to remove all those things which are non-coding into it, right? And then join on coding regions into it. What is that process called? RNA splicing, right? So mature messenger RNA is to be prepared, and for which you have RNA splicing. Splicing का मतलब क्या होता है? Splicing is cutting, right, and joining again together, right? So you splice the RNA and put it together, so it will become mature messenger RNA and mature messenger RNA. What happens now? DNA is always in a nucleus, right? DNA is always into a nucleus, right? This is what? A nuclear core, a bilayer core, right? And these are pores, so they are nuclear pores, right? Inside there is a DNA, right? Inside here DNA is there, right? From that DNA messenger RNA is made. So messenger RNA is made here, like with what happens to that messenger RNA? How it is always directional, right? 3 prime, 5 prime. So you have poly A tail into it, mature messenger RNA, right? Does not have poly A tail, right, into a DNA, but the DNA is just transcribed into RNA, messenger RNA, that is pre-messenger RNA. Pre-messenger RNA is spliced, poly A tail is added, everything happening where? In the nucleus. Remember, in the nucleus. From the nucleus, matured messenger RNA comes out, right? So mRNA, this is mRNA that come out from the nucleus. Now this is mature, it has all as poly A tail, right? They have its own cap region, right? So you have all the directions. Then this messenger RNA is taken to what is known as a small unit of the ribosomes. The smaller unit of the ribosome. So smaller unit binds first, right? And then the larger unit binds that one, right? On top of that, right? And then what will happen is the tRNA will bring one amino acid here. It is being what is being read is a code. So what is there onto the messenger RNA? The code, that is where the information is, and then the code is then decoded. Decoded with what? With tRNA. So tRNA has an anticodon loop. I'm sure you are aware of the tRNA structure, right? What's the structure of tRNA? It looks like this, right? It has a CAA region, CAA. region, that's where an amino acid binds, right? Then you have a loop, right? Then it goes an anticodon loop, then you have a side arm, right? Another loop, and then region. So it has a typical clover leaf structure of a tRNA, right? So tRNA, and then it binds to an amino acid here, which is an AA, that's amino acid, and then it goes and taken into the ribosomes. Now this anticodon loop, you have 3 bases there, right? And 3 bases on messenger RNA required to be matched. If they don't match, this tRNA doesn't transfer its amino acid, right? It's only after the tRNA reads the messenger RNA code, then only it will be transferred. Then this amino acid is transferred to the larger unit of ribosomes, and it is larger unit of ribosome that has the catalytic capacity. It will make the polypeptide bonds. So one amino acid from one tRNA, right? Another amino acid from another tRNA, third, fourth, fifth, sixth, seventh, and then it goes on like that, and then finally a chain of polypeptide is being made, right? Each joined together with a peptide bond. That's why it's called as a polypeptide. Polypeptide का मतलब क्या होता है? It's a many peptide bonds together, so many amino acids together, and then the pre-protein is formed. Now this pre-protein also has to undergo post-translational modification, right? Not that every protein is active at the same time. As soon as it is being made, it has to undergo post-translation modification, and then finally it will become active, right? So there are those things are involved into it. So let's understand what is a code into it, right? So what we know as code, right? So what is code, right? It is the information stored into DNA, right? Now I want you to take out your phone, right? Type on Google genetic code, and you will get a table there, right? Maybe you take an image search, so I'm sure you will get an genetic code table, right? What is a code table? It is given, I'm sure there are like A, T, G, C given here, right? In one, in another thing you have A, T, G, C given, and in the third line also there are A, T, G, C, A, T, G, C that is being given, right? And how you have to read A. So the first thing is A, right? What is the second? AAT. The second goes AAT. And if you go it likewise, the genetic code is triplet, right? So the important thing, what about that genetic? There are features of genetic code. We'll look into those. So the first important feature of genetic code is what? It is a triplet code, right? I want you to open your book and write down these features of genetic code, right? Number 1 is what? The code is triplet, right? Who discovered this code and how it was like been discovered? So you know that 1953 the helix was discovered by Watson and Crick. Watson was involved into the generation of and then explaining. See, Watson and Crick's job is not just to propose a model, right? It is not like that, but propose the functionality of the gene. Why we sing so much for Watson and Crick? Not just for the model, not just for the double helix, right? But how the double helix is going to function. So Watson was involved into so much of research even after then, right? For almost a decade of influence, right? You would get the entire genetic code and the central dogma was developed, right? In around 1963, right? So it's like almost 61, 62, 63, almost 2-3 years. But so that almost 1 decade has passed after the discovery of double helix to know and finalize the central dogma, right? So, so there are many people who are working together to delineate the genetic code. Marshall Narenberg is one of those, as well as Hargobind Khurana. Right. So we should be proud that one scientist, at least of an Indian origin, was involved in delineating the genetic code. Right, at those times, right, and of course he received, Hargobind Khurana received Nobel Prize for it. Right, you know that. Right, so they delineated code, right, and then this code, right, into that table what is given? AAA codes for what? There is an amino acid given there.
LYS.
What?
Short form LYS.
Leucine. Lysine. Right? So you have it will be given. Right? So what are those? There are 64 codes, right? Are there? You know? How many codes are there? So now the second point. The code is triplet के बाद क्या लिखना है? The second thing is there are 64 such codes. Why 64? Right? Because 3 raised to 4. 4 bases, right? ATGC and triplet code. So 4 cube is 64. So that is where 64 codes are there. Now 64 codes will code for how many amino acids? 20 amino acids. So that means what? Many codes are there for a single amino acid. And that is what is called as redundancy of the genetic code. So genetic code is redundant. Right? Or it is called as the degeneracy of the genetic code. Whatever is the third point under genetic, the code that you are going to write, the genetic code is degenerate. Right? Because of redundancy. Redundancy क्यों? Because 64 codes are there and they code only 20 amino acids. Right? They are redundant. But the next point क्या लिखोगे? Fourth point, right? Or whatever the number is, fourth I suppose. They are non-ambiguous. Non-ambiguous. Right? AUG, it will code for methionine. Right? And it will never deviate. So AUG will always code for methionine. है ना? कभी दूसरा नहीं आएगा. Right? So there is nothing, there is no non-ambiguous. Non-ambiguous मतलब क्या होता है? Look at the genetic code. There are specific amino acid given for that particular code. So ऐसा कभी नहीं होगा कि that this code will code for something different amino acid. नहीं. Right? They would always code for that given amino acid for it. Right? So that is where the genetic code was made. You know how the genetic code was delineated? How they discovered that, okay, that AAA would code for this and that AAT will code for that and so on, right? So forth, right? That's because they discovered a cell-free system of transcription, right? Translation. So they have a cell-free system. So you have ribosomes, right? And whenever RNA you will put into, right? And it will utilize certain different amino acids, right? So what they did, they made synthetic RNA molecule, all AAA, right? And then put into it, and they will see that okay, all protein now. So that means what? AAU code for protein, right? So UUU, if you would give all UUU messenger RNA, right? They will code for something. So all that is being done, and then they delete it. That's the work of Narendra, right? And cell-free system that is being developed by is one of those is Hargobind Khurana, who did it, right? That he discovered those cell-free systems for transcription, okay, to make the protein. And that is what Hargobind Khurana will meet him one more time when we will study PCR, polymerase chain reaction, because PCR is also related. It's also cell-free system for synthesis of RNA or DNA, isn't it? Right. So that will go there. Okay. Right. So understand that. So that is a cell-free system they use, and that is where they could delineate these groups. Right. So okay. So now the next point in the code, right? Characteristics of code are they are non-overlapping. Non-overlapping मतलब क्या होता है? If if it is like, say suppose this is a sequence A T T G A C C T, right? A right, this is a code, so we know it's a triplet. So what will it will code for? This first triplet, the second triplet, the third triplet, and then this is the last triplet. That is how it will go, right? So the first thing is ATA, right? TTG, ACC, TAG. Likewise, the DNA sequence go. Now look at the table. Tell me what is an amino acid coded by ATA? Which amino acid it will go?
It's
Isoleucine. Right? It will be isoleucine. Then TTG will code for what? See that TTG, what it will code for? TTG. Leucine. Okay. So isoleucine, leucine. Now GAC. No, ACC. Sorry, ACC will code for what? Threonine. Okay. Threonine. And TAG code for what?
Stop.
It's a stop code. It's a stop code. Right. So now what does that mean? Meaning what? ATA will always code for isoleucine. मतलब क्या हुआ? नॉन एम्बिग्यूअस, है ना? नॉन एम्बिग्यूअस के बाद क्या होगा? Then we have TTG will code for leucine. Now can I say that okay ATA for isoleucine? Now it will say ATT. Can I say that? No. Why? Because this A is already part of this codon. Right? Code is a plural. Singular is codon. So once ATA is one codon, this A is not to be utilized for the next one. So next one is what? The next three bases, and that is what is called as a non-overlapping. Understood what is non-overlapping? Non-overlapping का मतलब क्या हुआ? ATA के बाद I cannot read TAT, है ना? Then these two will be overlapping. Right? So once I say read ATA, the next three, and the next three, and the next three. Right? So you cannot utilize this T here or this A into this code. Understood that? Right? So that what does that mean? That means the code is non-overlapping. समझ में आया non-overlapping क्या होता है? Right? So it is non-overlapping. The another interesting part of the DNA code is what? That it is actually portable. You know what portable means? Means, say suppose I put this DNA sequence, right, into an E. coli, right, or I put this sequence into yeast. What will happen if I put it into E. coli? What sequence it will code for? Same isoleucine, leucine, threonine, and stop codon. Right? Even if I put it into yeast, the yeast would also do the same thing. And if I put it back into human, it would also be coding for the same amino acids. That means what? The DNA has got a portability. So that means what? In terms of genetic code, what you are going to write? The code is universal. The code is universal. इसका मतलब क्या होता है? Universal का मतलब क्या? Wherever you put this code in, it will be coding for the same thing. है ना? That's very interesting. That's why we can make human insulin into E. coli. है ना? So if we could like do all those genetic engineering experiments, like where we put human insulin gene, put it into E. coli or yeast or whatever system you are preferring to, but it will make human insulin. What we will call as it will still be produced, even if it is produced by E. coli, it will be a human insulin. है ना? So that's where you go to the medical shop, right, and ask for insulin, right? What is the name given to that? Humulin, right? That is human insulin, right? So, but then is it like that? It's extracted from human? No, it is been gene being taken from human, put into E. coli or yeast, and human insulin is produced. So even if yeast is producing, it will produce human insulin. Why? Because it is using a human DNA, right? And that's why नीचे लिखा हुआ होता है ना, it's like a rDNA product, right? What is an rDNA product? It's a recombinant DNA product. Recombinant DNA product means the DNA from human is being taken and put into yeast or E. coli, right, to or a bacterial system to make human insulin. है ना? So one important thing क्या है genetic code का? That it is universal, the universality of the genetic code. है ना? Right? So those are the genetic code features, right? These are important genetic code features. So those code are there into DNA. See, the important part of the genetic code or the central dogma is that it explains so many different things to you, right? It explains many different things to you because of this. See, suppose you want to change something into a strain. So there is called as a strain improvement. You want to improve a strain. What you will do, right? You will not change the protein, right? Neither you will go and change the RNA. But what you will do, you will change the DNA, right? So if you change DNA, it automatically would translate into RNA, right? And RNA would automatically translate into right. So if you are even if you want protein, recombinant protein, right, you are not going to try and change protein. You are not going to try and change the RNA. But what you are going to do is change the DNA, because that's a stable change. First of all, right? Once you change the DNA, it will be there forever, like change in the change condition, and then it is inherited. So when that E. coli is going to, suppose you are modifying E. coli genetically, right? So whenever that E. coli would replicate, all the progeny will contain the changed DNA. That is most important part. And then when they change DNA, it will reflect into proteins. Well, the same thing happens in case of the genetic disorders also, genetic diseases also. If I have a change in DNA, what is called as a mutation. So if I have a mutation in DNA, I will have mutated RNA, and automatically it will reflect into mutated protein and mutated function of that protein as well. And most importantly, then I am likely to transfer that mutation to my offspring. Right? So that goes inadvertently, but then the genetic flow is into this direction. Right? So the central dogma tells you what? This is the way genetic information flows. Right? One more important thing is the genetic information flow of genetic information is unidirectional. That means what? It will not go from protein to RNA or RNA to DNA. Only exception is it can go to RNA to DNA, right? In case of, we say this transcription and translation. There is no reverse translation, but there can be reverse transcription. RT, right? That is what we call as RT, right? Reverse transcriptase PCR, right? Or reverse transcriptase. All RNA viruses, right? We have talked about it. All RNA viruses, right? Although their genetic component is RNA, they convert it into DNA in order to infect, right? The classical example is the HIV virus, right? It's an RNA virus. Even if it is an RNA virus, it will convert itself to DNA and then infect, right? So it has to undergo that. So barring this part, the entire genetic flow of genetic information is unidirectional, right? So it doesn't go from this to that back again, right? So from protein you cannot make RNA, but from RNA you can make DNA, right? Understood that? Right? Clear? This is clear? Okay. Now you have understood the genetic code also as well, right? And with the help of genetic code table, you will be able to delineate the code, right, to the amino acid what they code for. Most important part, you have to understand that. Again, go to the genetic code. There is one thing written into red, AUG, right? That is methionine. What is AUG? Right? Or ATG is a start codon. Meaning what? All ATGs are start codon, and then you will find that there is no redundancy there. Right? AUG only codes for methionine, and methionine is this is the single code, right? That methionine used. Right? There is no other code for the methionine, so there is no redundancy as far as the methionine is concerned. Right? So all your proteins are going to start with methionine. Right? Most eukaryotic protein therefore begins with methionine. Right? So so obviously methionine also become like a start amino acid, right? Because its code is a start code. Right? Many proteins that you don't see methionine as a start amino acid are post-translationally modified. Their caps are being cut off. Right? There is a middle sequence that is being cut off. Right? So you will see a protein without methionine as a start. There could be a methionine inside the cell, inside the protein. Right? It's not that okay, it only exists at the beginning. There is methionine inside in the otherwise sequence of the protein too. But then mostly proteins would start with methionine. Right? And then there are 3 stop codons. Right? Pay attention to that. Stop codons. Which are stop codons there?
UAA, UAG, UGA.
You could say UAA, UAG,
UGA,
and UGA. Right? This is what the stop codons are. So stop codons. What does that mean? Why it is a stop codon? Right? Why it is considered as a stop codon? That's where the chain would grow and stop. Why the chain would stop there? That's because there is no tRNA that would recognize this sequence. Right? So as soon as this code will be there is no tRNA, so there is no anticodon for that. Right? What will happen if we make a tRNA synthetically, which would recognize this and carry some amino acid? Then it won't no longer become a stop codon. Understood that? Then it will not become a stop codon. Why? Because there is no tRNA for this. That's why they serve as a stop codon. Right? Because there is no tRNA. No tRNA means no amino acid. Right? So the polypeptide chain terminates there at the stop codon. Understand that? So try to understand this, and then it is being stop codon. Pay attention to, say, suppose leucine. How many codes code for leucine? Let me know that. Lysine देखो चलो, lysine के कोड बताओ मुझे. Lysine के कोड बताओ. How many codes are there for lysine? 2. Not just 2. ल्यूसिन के कितने हैं? 6. ओके, चलो ल्यूसिन के बताओ. Now look carefully, right? ये एक है और ये दूसरा है ब्लॉक. ये ऑल कोड फॉर ल्यूसिन. Barabar? Right. Now look here. The first code will always be C. यहाँ पे भी C है, यहाँ पे भी C है, यहाँ पे भी C है ना? In all four, the first letter is C, the second letter is U, the third letter is
All four.
can be changed. Right? Even in this case, the first two letters are U, U. Only the next letter can be changed. U, U, the next letter can be changed. Right? So this is called as a wobble hypothesis. Meaning what? When the tRNA will come, it will recognize these first two codes. So although we said the code is triplet, right? The first two letters in the code are the most important. They are the determining thing. The third can be anything. Right? If you look into all other amino acids which have multiple code, you will see the same thing. Right? So what does that mean? That means the third letter can be a wild character. Like when you play the card game, right? You know that joker you can put, right? Which is a wild type character. It can take any place. Then the same thing here, right? CU is compulsory, but there could be an optional on the third letter. So in the three-letter code, first two letters are mandatory. The third can be a wobble, right? And that is what is called as a wobble hypothesis, right? Or that is called as a degeneracy of the code. So what you are going to write in the code characteristics? What is the next number? Fifth or sixth? Seventh. In the code characteristics of codes. The seventh is the codes are wobble, right? Or they are degenerate. What do you call as? Degeneracy of the code means the third letter is degenerate, the first two letters are increase. Clear? Right? So in the genetic code, what's going to happen? The third letter is anything, right? But the first two letters are important. So the genetic code, the first two letters determine what amino acid it will be. The third is not so important. So it is a degeneracy. So what happens? What is the problem here? From DNA, you can definitely guess the RNA sequence. Right? From DNA, you can definitely guess the RNA sequence. But if I ask you to from RNA, if I give you RNA sequence, and if you try to guess the DNA sequence, what will happen? Then it will be a complicated. What will happen because of that? If it is CUG, it can be CUA. So it will become a degenerate. So from the protein sequence, if you try to guess the RNA sequence, it will become difficult for you to do that. From a protein sequence, right, you cannot guess the DNA sequence. But from DNA sequence, you won't be able to make protein sequence. Using this code table, what you can do? From the code table, you can easily make, if the given DNA sequence, if I give you a DNA sequence, like we have done it here, a DNA sequence was given, from which we can guess the protein sequence. But if we have given leucine, isoleucine, threonine and stop codon, and then ask you to make DNA, there could be 3-4 versions that it will be. Right? From isoleucine you will have many codes, so you cannot make a guess of the DNA. So because of the degeneration, that's why it's called as the code is degenerate. Okay, understood that? Right? So what we talked about today are the central dogma of molecular biology. It's the flow of genetic information from DNA to RNA, RNA to protein, right? And I extend that central dogma to protein to the metabolites and metabolites to a trait. That is how it will be. So protein to metabolites and metabolites to trait. So if you accept, but a classical central dogma is only this much: DNA to RNA, RNA to protein. Right? And DNA can replicate. So that's why that circular arrow is given on top of the DNA. That is a replication. and then this is the central line. Okay, right? When we talk about genetic information, what happens is the information in the DNA is in the form of triplet code, right? And then we saw that 3, 4, 6. How many it becomes? 7 characteristics of the genetic code, right? The genetic code is triplet. It is non-redundant. It is non-ambiguous. It is non-overlapping. It is universal, and it has degeneracy, right? And then every word you could understand now, right? That what happened with the genetic code. Okay, right? So I think we'll stop here for the today, and then we can take the next thing in the next. Okay.